have the first of two guest lectures dealing with issues of basic biology, and then on Wednesday I think some additional comments and a discussion of how these biological principles relate to the issue of cancer study and also cancer treatment and so on. And our guest speaker today is Dr. Ana Corbacho, a member of the education team here at the Biophotonics Center, a good friend and colleague who has her PhD in the biological sciences and has a lecture that by 10 minutes before noon we'll all have a basic biology vocabulary. The floor is yours.
Good morning, everybody. So I got the task of teaching basic biology in 2 hours, and I had to kind of do a very general thing, but still give you the principles of what biology is. So I'd like to know first, what is my audience? How many of you have bio background? Okay. Biology background? Engineering? Physics? Math? What is the rest that are not raising your hands? What are you? Or
like our major?
Yes.
Or major.
Well, major and knowledge about. I mean, have you taken biology in high school? All of you.
Yeah.
All of you. Okay. Okay. So at least we can start from a common ground. Yes. So the first thing that I'd like to talk about is, okay, and I'm starting from a very general point of view, that is, okay, where all this study of biology started and how it got organized. That kind of explains where are we today and how we got there. So first of all, we have a lot of diversity that here is just what we can see with the naked eye, but there's a lot more than that. And then how we get to organize that, to study it, and to understand how it works. And the first ideas, can you see that? The first ideas about organizing this, for some reason the color is a little bit... Oh. Okay, we'll see. The first ideas about organizing what you can see were with Aristotle, and is the one that started the field of taxonomy. A second classification came in the 1700s with Carolus Linnaeus, and he classified all living organisms that he could recognize as plants or animals. The ones were green and didn't move were plants. Everything that moved and was not green were animals. So basically was what he could see and how he could classify that at that point. Later in the 1960s, we have Whitaker defining the 5 kingdoms of life. And we have Protistas here, or Monera. Monera here in the bottom. Protistas here, unicellular organisms, but eukaryotic cells. We have plants, fungi, and animals. That was in the 1960s. And all these classifications starting so early until the 1960s were based on the phenotype. What is phenotype?
The genes that are expressed.
That's genotype. What is phenotype?
That's the physical manifestation.
The physical manifestation. So it's basically the characteristics that we can see. Color, size, it moves, it doesn't move, it eats, it doesn't eat, that kind of thing that we can see directly. So we're characterizing something on what we can see, that thing does. So basically, until the 1960s, that was the kind of thing we had. We had microscopes, we had all kinds of things, but we were just watching the characteristics. Now, with the advancement of biology and the era of molecular biology, things started changing a little bit. And then we have, in the 1970s, with Huss, we have the classification based on the genotype or the DNA. So now we are able to determine the genetic code of all species. We are able to read and see how they look alike or not based on the DNA, not anymore in the external characteristics, but in the genetic code. And that classification now puts all these in a different shape, where we have three big domains of life: the archaebacteria, the eubacteria, and the eukaryota domain here. Now, in each of these domains, we have a lot of other kingdoms. These two are bacteria, and these two are all eukaryotic cells. So before we go there, what are the characteristics of things that are alive? How do you know that something is alive? What is the definition of life? Wow.
Units life energy.
Energy. It needs energy, and that is in a living organism is in the form of what molecule? Basically, ATP. What other thing? Many things need energy and are not alive, or generate energy and are not alive.
Create more life.
It can create more life. So it would be the idea of self-replication. What other thing? Responses to stimuli. Response to stimuli. So you touch it and it does something. Okay, even if it's a plant and it doesn't move, if you do something to the plant, there will be some kind of response. One more. There are computers that do that, right? There are dogs that are computerized that if you touch them, they do something. So that's not enough to be alive. It's part of it, but it's not enough. What else?
They have cells.
They have cells. So all living organisms are cells or are formed by cells. So a cell is the basic unit of life, of everything that is alive. Okay, and let's go to the classical list here about the things that are necessary to consider that something is alive. First of all, living things are composed by cells. It's the basic unit of life. It could be unicellular or multicellular, a multicellular organism. But they all have to be, the cells have to be there. Second, they require energy, ATP. Somebody mentioned that. They reproduce, and they are able to self-replicate and form identical progeny after it. It displays heredity. So there's DNA in all these living organisms. They respond to the environment, respond to stimuli, somebody said that there, maintains homeostasis. So this is a characteristics where the organism is able to respond to stimuli and maintain certain balance. It's like you have a cell exposed to certain chemical situation and is able to kind of, to a certain extent, maintain the internal balance of it. And depending on the temperature may change and everything, but still the organism is able to get certain equilibrium depending on what's going on. There's the homeostasis of self-regulation are kind of the same thing. And then finally, they evolve and adapt. Living organisms adapt to their environment and are able to evolve. So those are the, there are a lot of discussions among scientists about what does it really mean to be alive. And these are the main minimum characteristics that everybody agrees on. That are really characteristics of living things and not other things. What are the, yeah?
But if self-replication is one of the requirements, like what about animals that can't reproduce, like mules, things like that? Are they technically not alive?
No, they are alive, obviously, but there are conditions where. That's a good question, but no. But realistically, it's alive. There's a condition. It's not like people that are sterile are not alive, obviously. So it's a condition that somehow it's affecting the system in a way that doesn't allow the mule to reproduce. But it's not. And the mule is a cross. So in its own species, it's able to self-replicate. When you cross species, you are generating situations where the systems are not working completely well because you're crossing. But that's the kind of situation. So what are the things that all these living organisms have in common in terms of the biochemistry? There are 4 biomolecules that all of these organisms share. DNA, nucleic acids, because it's DNA and RNA, lipids, proteins, carbohydrates. Those 4 are the basic 4 biomolecules. And I'm going to go in a very general way through all of them. So then lipids. What is the basic function of lipids in a cell? Is to make membranes and establish compartments. And the way that they do it, so the membranes create the walls between the different compartments. You have the basic lipids that form these membranes are phospholipids that have a polar head and a non-polar tail. And the way this is organized is in B layers or micelles. The B layers in a way that the polar heads are towards the water and the non-polar tails are towards the inside. So in a way, these phospholipids, because they have a polar head and a non-polar tail, they're able to self-assemble and self-organize, either forming this shape, but this shape is not useful in the cell because it's just a little ball made of, with no water inside, made of phospholipids, and then a B layer that is the basic structure of all cell membranes. And then in the cell membrane, of course, you have proteins going through and all kinds of modifications of lipids that generate other functions. Nucleic acids. We have 2 basic types of nucleic acids, the DNA and the RNA. And as you know, the DNA are formed by 4 nucleosides, adenine, cytosine, guanine, and thymidine. And DNA contains the genetic information that encodes protein. We're going to go into detail next week. We have a molecular biology class, so this is just a little bit of a refreshment thing. Proteins. What can you tell me about proteins? They are formed by amino acids. There are 20 amino acids, and what do they do in the cell? They work as enzymes, so they catalyze chemical reactions. What else? They regulate the cell, like for example, they are the ones that do the signal transduction stuff. They allow the cells to communicate between each other. You said that? Exactly, because you have receptors that can, first of all, take a signal that comes from the outside and internalize that signal that goes into a signal pathway into the cell. All those are, all that chain of reaction is made by proteins, different proteins with different functions. So a protein can be a signal that comes from the outside. There's a receptor that accepts that signal, and there are proteins inside that translate that signal inside. So signal transduction for sure. What else? Chemical reactions, enzymes. They can protect the cell. Yes, an example? like antibodies. In that case, it would be a signal molecule also in the external side of the cell. What about the structure of the cell? The scaffold, the cytoskeleton is made by proteins. So basically in the cell, everything that works, everything that moves, everything that is doing something, in most cases are proteins. They are all formed by 20 amino acids, but the function of that protein will depend on the sequence of amino acids, the structure of that protein, but it really can do, proteins do absolutely everything. The DNA alone, without the help of proteins, can't do anything at all. If there are no proteins that come to the nucleus to open the DNA and read it, nothing happens. So here there's an example of protein that I like to mention just because you've probably seen it before in terms of biophotonics, and how is hemoglobin. And hemoglobin has different color if it is combined with oxygen or if it's combined with CO2. And I think Marco mentions this in his lectures. When you see the pictures, the anatomy pictures in the books, you see blue and red, blue for vein and red for arteries. And the real color is this. As you can see, this is a little bit dark, purplish, but it's not even close to blue. And this is fresh blood combined with oxygen. That's how it looks. And basically, using light, using this difference in colors, we can measure a lot of things. But you can get to see it when you put them together. Carbohydrates. Why do we need carbohydrates? I have two examples there. Glucose and glycosaminoglycans. Glucose, we all know, energy. Okay? Without glucose, we cannot generate ATP. What about glycosaminoglycans? Have you ever heard about this? These are very large carbohydrates. This is a monomer, and these are polymers. These are polymers. This is an example of how the cell uses different monomers to create these huge structures that are outside of the cell. And these things outside of the cell, what they do is help in communication cell to cell. They are able to maintain the extracellular liquid with certain structure. They are able to kind of store some factors and hormones. So basically, they're outside, they hydrate, and they form this kind of gel outside of the cell that allows for all these things to come and go. Imagine you only had water. How do cells get better only if they have water? They have to be really attached. And there are some cells that are totally attached to each other, like the epithelial cells in your skin. But there are other cells that have a little bit of separation between them. And the glycosaminoglycans are in between, generating that kind of buffer area for things to come in and out. So we went through a little bit what is the idea of what's a living thing, what are the chemical blocks of the living things, these four biomolecules. And now I'd like to talk a little bit about the different cell types. What two main cell types we have? Not we, there are out there.
Prokaryotes and eukaryotes.
Prokaryotes and eukaryotes. And here we have the representation, very basic representation of a prokaryotic cell and a eukaryotic cell. And the main difference between them is that prokaryotic cells are very simple. It's like a little bag of cell membrane with DNA inside and very minimum specialization. And they're small exactly because there's not too much capacity to specialize. And the eukaryotic cells have a lot of internal membranes, a lot of compartments that allow for organelles. And they are larger because now they can organize all the chemical reactions in compartments. So now we can have many more chemical reactions and many more things going on because it's compartmentalized. There's a limitation of how many things you can be reacting in the same compartment because you're keeping the same conditions. The pH is the same, everything is going around, some enzymes may interfere with the others, But when you start having compartments around, you can separate things. Then you have a mitochondria. A mitochondria can be doing all the reactions at a certain pH that is different from what the nucleus needs. Okay, so that kind of compartmentalization allows for much more specialization in the cell. So one thing I want like you to keep in mind from now on is the difference in size. Here you have a scale, and a bacteria goes from 1 micrometer to 10 micrometers approx, and then eukaryotic cells start at 10 micrometers and go up. Most cells in mammals are between 10 and 100 micrometers. What is the largest cell that you know? An egg. What type of egg?
Human, female.
No, that's tiny, tiny.
Oh, like ostrich egg?
Ostrich egg. The fertilized ostrich egg. And which part of the egg is the cell? So you have the shell, you have the albumin, you have the yolk. What part of the egg is the cell? The yolk. The yolk. So as you can see, the level of specialization that eukaryotic cells could reach, because they started the compartmentalization process, is huge. You can have massive, huge things going on. The idea of this class is not to go in detail about the different organelles in the cell, but we can go over it. While in the prokaryotic cell, we have just a little sac of membrane with DNA, 1 chromosome of DNA all loose in there. In the eukaryotic cell, we have a nuclear envelope that encloses the DNA in the nucleus. We have the endoplasmic reticulum, the rough and the smooth. We have mitochondria. We have Golgi apparatus, lysosomes, and we have membrane specializations in the surface. All kinds of things that have particular functions and particular structure and particular proteins that do certain functions, and that happen only in that compartment. Do you have any questions so far about this? So now with that said, and in terms of size, what are the kind of things that we can it's in terms of what are the kind of instruments that we can use to observe things. In the case of bacteria, we said it was very small, right? Here we have these two domains are bacteria. There are huge amount of bacteria in the world. And when we are using light microscopy, we can see to a certain extent, and this is the maximum up to here. This is light microscopy. To see further than this, we need the electron microscopy. All this area is in the electron microscopy field. So this part, bacteria are alive. From here to there, we have to kill the bacteria, fix them, cut them, prepare them for the electron micrograph. Okay, so light microscopy in this area is alive. In this area, it's dead, to be able to see what's going on. Now, when we go into eukaryotic cells and eukaryotic organisms, we can have unicellular organisms or multicellular organisms. When we are talking about unicellular organisms, we are talking about a level of compartmentalization and specialization that will change according to what is the subject we are talking about. This is an example that I chose for you to see because it's one cell, and you will see it doing a lot of different things and responding to things. It's called vorticella. It has a structure that attaches to something that doesn't move in water, and it's retractile. It has, you see different organelles inside, and there's an area here with cilia, this little, you see this little hair here moving? These are cilia, and what they're doing is moving the water around to be able to attract and trap food. So it's one cell, and it has different parts that are very specialized. It has a mouth, it has a retractile portion. It's able to detect that this is moving and is retracting to protect itself so that the current doesn't get it out and get it lost in the big stream. So it's only one cell and it's able to do many different things. Another example, and I'm showing this just for you to see the level of specialization we're talking when we're talking about a eukaryotic cell. This is another type, see this here moving? These are cilia, all these organelles inside. You can see the nuclei inside. These are microorganisms found in fresh water, in any stream. Another example. This is a paramecium. This is the mouth of the paramecium. Again, you can see these little hairs moving around that those hairs allow the paramecium to move and go wherever it wants. Basically, this cell is covered with cilia all over the place and can rotate and can move back and front and can do all kinds of things. And you can see the little organelles here. But it's amazing the level of specialization that one cell can have. It can figure out what direction it wants to go, if there's light or there's not enough light, if the water is dirty or not, it will retract, it will move away from substances that they don't like. They can trap things and eat things, and they can dispose all the material that they want to excrete. So when we're thinking about an organism like us, it's like, yeah, obvious, but when you're thinking about a cell, and it's able to do all that, it's pretty amazing. So as you can see here, you can all look at the structure and what's going on with light microscopy. And the organism is alive. We don't need to kill it to see what's going on. We can observe. Now, what happens when we go into the multicellular level? Here I'm going to show a simple organism, it's a mold. But it's starting the multicellular level, and we have already rudiments of tissues. A tissue is an aggregate of cells that have the same function and have similar structure. So here we have mold, and again, light microscopy, mold is alive, no problem. And here it's alive all the way. Okay, we're able to see here the two different tissues that the mold has. The mycelium, that is formed by cells that form branches, but there are septum between them. As you can see, one cell, another cell. And then the reproductive tissue that is formed by spores. It's only two types of cells that you have there. But already you start seeing the rudiments of tissues. And up to this is fine. We can still see everything alive, and we don't have to kill it to see most of what's going on. And basically, with this, we have reached the limit of resolution of light microscopy in terms of what we can see. Here we have the spores of the fungi, and there are things moving around. Some of them may be bacteria, like these little things moving around. There's debris also, but this is the size of bacteria in comparison to the spore of a fungi. Now, basically, we can see this because it's transparent and because it's isolated. And the level of multicellular organization is still very rudimentary. There's not much going on. What happens when we get there? When we get to complex multicellular organisms, we start having tissues, real tissues, that organize, forming organs and forming systems. And keep in mind where we're talking about what can we see with light microscopy and what can't we see. So you've seen this a million times, and this is just a way of organizing what we have. Of course, all our systems are connected together. The muscle system or skeleton, the skeleton will not move without the muscles. The muscles will not move without the nervous system. It's all connected, but the way we study is separating things. And here, there's a level of, what are the levels of organizations of higher organisms? We start from the molecular, the chemical level. We go to a cellular level. Then we get a tissue level where similar cells with similar function get together to form tissues. And then tissues organized in systems that serve one type of function or one group of functions that are similar in the organism, and then all together form the living system. Okay, so what with that? What happens now? What can we see in this organism using light microscopy? If I give you a microscope, how much can you see of that living organism? If I give you a microscope and tell you, look at something of yourself in that, what would you put there? A what? Samples? A biopsy. So now you have to go in and cut. Yes, you have to go in, cut, fix, slice, stain, so it's not alive anymore. But if I give you a microscope in the class right now, what kind of thing can you see? It's not much you can see. Not many things. You can think about things like a little bit of skin if you have luminescence, so you have light coming from the top, but it will not go through. So you can see a little bit of skin. You can put your hair. You can put cells from your cheek. And we're running out of things. You can put sperm, but basically we're running out of things. Well, you can poke yourself and put some blood, but we're there. There's no way of using a light microscopy and seeing anything alive and functioning in a large organism like us. And that is where biophotonics comes to play. Finding ways of having an organism alive and being able to see what's happening without killing it. And an example of how you will look at things, for example, normally what you do is you fix, this is a lung of a mouse, you fix it, you slice it, and you stain it, and you're able to see up to this level, to the cells there, and a little bit of the tissue there, blah, blah. But then to see at this level, or what's going on at that level, you need, again, the electron microscopy. All the way here is dead and fixed. Just because the amount of cells and tissues that you have doesn't allow the light to go through and observe what's going on. You have too many layers of things. You can't see what's going on. That's a huge difference in comparison to simple organisms where you can go and put them under the microscope and observe what's going on. In larger organisms, that doesn't happen anymore. And on the top, you have blood that will interfere with light absorption. You have a lot of colorful things that will interfere with that. So that is a big limitation to study what's going on in a living organism. And I'd like to now use the last couple of minutes of this part of the class to go into an example on how you would use electron microscopy to determine what's going on and what is the kind of thing that you have to do to be able to see really what's going on. And I'm going to use an example of this little hair. This little hair have exactly the same structure than the hairs in those unicellular organisms that I showed you. They're called cilia, and the structure is exactly the same. If you take a protein from one of those protists organisms and you substitute it by a human, same kind of protein, it will work. The structure is exactly the same and is conserved along the evolution. This is just to show you that in this case, when we're talking about bacteria, from here to here is alive, here is dead. In this case, it's dead all the way. And now I like to, as I said, I would like to talk to you about the cilia. And this is a cross-section of human cilia. You can see that you have two central tubes. And then you have 9 pairs of tubes in the forming a circle. And you can see these little arms coming out. Can you see that? Those are called dynein. All these structures are made by protein, are called microtubules. Those tubes are called microtubules. And the legs that are coming out, or the arms that are coming out, are called dynein. Dynein is a motor protein. So you have tubulin that forms the tube that is structural. It's forming tubes. Everything is protein. The dynein that comes out is a motor protein that will use ATP to move, to attach to the next tube and move the structure. So basically, imagine you have these tubes all together, and then you have arms that come out and do this and attach. So while they attach and they move the parallel pair of microtubules, the cilia beat. It's able to do this kind of thing. And all the cilia in the surface of the respiratory epithelium move at the same time. They move in waves. And the reason why they can move in waves from one cell to the other one is because all the cells that form that epithelial layer are connected by channels that allow ions to go through and let the whole layer know that, okay, now we're moving this way, and now we're moving this way. So basically, it's acting in an organized way, even when we are talking about different cells. In the case of the unicellular organisms, you saw the cilia moving, and you know what it's doing. Okay, it's beating, it's moving, it's allowing the organism to go from one place to the other one. It's allowing the water to circulate, so they trap food. Why do we need cilia in the respiratory system? What? Exactly. It gets all the stuff that comes in. Part of it will be trapped in the little hairs we have in the nose. Other parts will be trapped in the mucus that you have in the nose up to the larynx. But then after that, anything that comes through will get attached to the surface of that mucus that is covering all our trachea and bronchial. And how do we get it out? It's stuck there. And now what? It just accumulates until it builds like a chimney. No, it needs to come out. So you need a whole system organized to get those cilia beating up, going up until you get all the gunk out, all the mucus out. So all that dark stuff that comes out is because it needs to come out. Otherwise, it would stay there and it would form these big infection situations. And that's what happens when you have a mutation in this little leg that is DNA. If you have a mutation there, you're unlikely to be born with that. Your cilia will not move, or will move in an unorganized way. And what happens is exactly that. The gunk stays in, and then the baby starts having problems early on, has a lot of respiratory problems, and once it's diagnosed, the only solution for that is physical therapy, to find a way to get all that gunk out. You need to do some kind of physical thing to help that to happen. But that comes from one mutation in that little thing. And the way we can use electron microscopy in the old ways to figure that out, this is the structure of a normal cilia again, with the 2 DNA in and the microtubule doublet here. These are examples of real patients, real kids with respiratory problems, where you can see that there are different types of situations. What I just told you is the kind of mutations in the DNA that may allow, first of all, to have kind of partial DNA happening, DNA going in the opposite direction. See, all these go in this direction, and this one goes contrary. If you have that kind of situation, the cilia can't beat in an organized way because they will stick in the wrong place and they will have opposing forces in the same microtubule. So that would not allow the cilia to move. So this is one of the common situations of mutations in DNA. Then we have situations where the organization of the cilia are not right. For all the cilia to move in the same direction, you need to have these doublets or microtubules in the same kind of parallel way, so they can all move this way. If you have them all crooked like this, they will move in different directions, so there's no way of organizing everything to go in the same direction. And then there are other cases where you have cilia where you don't have the central pair of microtubules, you only have one, you have three, you have a doublet in the center that is totally crooked, there's one isolated microtubules here, and there are all kinds of situations where mutations in the organization of these can cause a lot of trouble. So for seeing this kind of thing, you have to either do a DNA test to confirm that you have a mutation in the DNA. Not all the mutations will create this type of situation. And to finally diagnose this kind of situation, you have to do electron microscopy. So basically, you have to go into a surgery, take a biopsy of the nasal cone, and go do electron microscopy. Yes.
Are those mutations like widespread in all the cilia, or are they just?
They are in your DNA, and they are all over the place. So the gene that codifies for that DNA will be mutated, and then you have this defect all over the place. And there are cilia structure like in other places. Like there are cilia in the abdomen, so when you are developing and everything rotates inside to establish the systems in the abdomen, you need cilia to help rotate. Many of these kids are born with the organs in the opposite side, like not well rotated, and that generates other kind of problems. What about spermatozoids? Spermatozoids, the tail of spermatozoid has this same structure. So those kids are not going to be fertile either, because the tail of the spermatozoid will not move correctly. So this is just an example about how understanding how a protein works and the DNA and everything. And so we're combining the DNA with the structure of a protein with the electron microscopy to understand how a disease is playing a role there. So now this is enough for this part of the class. We're going to have a 10-minute break, and then we're going to go into the second part. Do you have any questions about this? I know it's kind of a big picture, but I hope I covered everything that is necessary for you. Okay, 10 minutes break.
I like this side thing. Is that are you not they go around. They go around.
I had to like move it, but they were like under the covers and all I could see was like, I was like checking it on the page. I was like, I don't think I like sounds like money, but my back is cold. But whatever. I only have one now. I got 3 for Christmas. 3? Yes. Yeah. I have 1. You were in your 20 last time?
I know. It's actually people coming to my floor. So like you peek around the room, they're cool.
So what about we continue? So that first part was my attempt to kind of cover all biology in 40 minutes. And the concepts that I'd like you to remember there for your exam and later, the things we got were ways of organizing living things. Second was 4 biomolecules. Prokaryotic versus eukaryotic. Basically here the idea of compartmentalization is what we mostly talk about. And then the idea of how the limit of light microscopy is associated with how many layers of complexity you have in an organism, and able to connect that. Why is that we have more limitations the larger the organism is? Okay? So then in the next part of the class, I'm going to try to bring to life a few of these concepts working together. And I named this part 2 successful stories of biology and chemistry. The first part of the talk, so the first story, is it's about using model organisms to study things. So here I will show what are the most classical model organisms that have been used in biology for 100 years or more. And we have some typical ones. In the case of viruses, we have the Lambda bacteriophage. It is an electron micrograph of the virus. At the level of bacteria, we have Eukaryotic coli, and I'm pronouncing in Latin, that's one is E. coli in English. So Eukaryotic coli. Again, it's an electron micrograph. Here we jump into, it's large enough to be seen in a light microscopy. And here we're talking about Dictyostelium discoideum, and it's a very rudimentary fungi type. Yeast, Saccharomyces cerevisiae. Have you heard about all this in some point in your studies? Sometime. In plants, we have Arabidopsis thaliana that is being used a lot for everything. when we are starting in the animal kingdom, we're talking about Caenorhabditis elegans, or C. elegans. A more evolved organism has been the fruit fly, Drosophila melanogaster. In vertebrates, in the lower level of vertebrates, we're talking about the zebrafish or Danio rerio. And then the mouse, mus, with the babies. So what do you think these model organisms have in common, and why people have chosen them to keep studying them over and over and over and over? I mean, they're model organisms. A lot of scientists choose this model to study for all their careers about a specific thing, protein or something.
They have short life cycles.
Short life span. So that means that you can keep them in the lab, that you can have, you can study something from one generation to the next one pretty fast. So all of them, you can, it's kind of, you don't have to wait for a tree to grow over 30 years to see what happened. You see it in 1 week or 2 weeks with a plant, and you can plant the next generation immediately. Yes?
Kind of trivial, but they're all really small.
Sorry.
It's kind of trivial, but if they're all really small, you can have a lot of them in the same place.
Yes, and it's not trivial at all. It's one of the most important things. They are small. So basically, you can have a ton of them in your study, and you can have thousands of plants, and you can have thousands of fish in a pool, and you can have a lot of individuals that will increase the end of your experiment, and then will give you a result faster. It's different when you are working with monkeys. In the way you design your experiments, if you're working with monkeys, you can't have an N of 100 monkeys to cut the stomach, because it's super expensive and it's not practical, and it has a lot of ethical issues associated. Okay, when we're talking about a lot of yeast, nobody has problems about the ethical issues with yeast, right? When we're going up in the scale, things get much more complicated. Fish, we can do a lot with, and nobody cares much. We eat fish all the time. The problem is when you're going up in the scale, things get very complicated, very expensive. So the size is very important. It's not trivial at all. Drosophila melanogaster, you can raise a lot of them in a tiny tubes with yogurt and nasty stuff there, very fast. What other thing do you think? Yes?
They're all genetically similar to each other. They're all similar to each other genetically.
They are all similar to each other because we share a common evolution path there, evolutionary path. So the genetic material has evolved, but we all share the basics of it. So the same kind of DNA and protein that you can find here, you can find it in the mouse, and you can find it in small organisms. These ones don't have DNA, but like I would think, like I can tell you, like an enzyme that comes to my mind, that like sodium nitrate synthetase, will be the same in all of them, the same structure. What other thing in these small organisms? I think I can go and move on into this. That we're going to find more of these characteristics in this slide. So I'm going to take from those organisms, I'm going to take one example, that is Caenorhabditis elegans, to illustrate all the other characteristics why model organisms are useful. In the case of C. elegans, it's been used a lot in these areas of research: genomics, cell biology, developmental biology, neuroscience, and aging. The reasons for being used in this area are exactly because of the characteristics that this worm has. This is a worm that is a free worm. It lives in the soil. It's tiny. It measures approximately a couple of millimeters long. It's tiny, tiny. You can see naked eye, but it's almost invisible, naked eye. It eats bacteria, so that makes it easy to feed in the lab. You have some bacteria they eat, that's fine. You can grow the bacteria in agar plates, so it makes it a very easy subject for having in the lab. Very small, as we said, short life cycle. It's about 3 days. Again, you have new generations happening all the time. They have a big progeny, and this is the other characteristic about all the models. They have a lot of babies.
So what's progeny? What is
Progeny is babies, like descendants. So they have a lot of babies, and that's the other characteristic. With the mouse, you have about 9 babies in each delivery of the mouse. So still, you have a lot. It's not like 1-to-1. Like primates will be 1-to-1. It's very complicated to work with that kind of subject. They're very easy to propagate, and you can freeze these organisms. You can freeze the eggs, and you can melt the frozen thing, and they come back to life. So these kind of situations are very unique and are very easy to work in the lab with. The other characteristics are that they are easy to manipulate genetically. They have a very compact genome. That this means the DNA, the amount of DNA is manageable. It's small, relatively small, and it's easy to manage. And the sequence, the DNA sequence, has been completed a long time ago. So the whole sequence is available for people to study for a long time. This was one of the first organisms that was completely sequenced. They have invariant cell lineage between animals. These worms have about 1,000 cells total. That's it. And they all have the same amount of cells in the same place. So basically, once you study how a cell came to be in that place, you can study for a long time until you understand what happens and how it happens. Because that cell starts being here with the mom cell and migrates in this direction, and here it divides, and it happens the same way in every organism. So you can study and study and study the same process until you understand it. Another big thing, this organism is transparent. You can go through. You can see through everything, and there's no problem. And finally, these organisms have 2 different sexes. There's a male that doesn't seem to be that useful. It seems to be a little bit of a dead evolutionary path in these organisms. And the other thing is that they are hermaphrodites. So they have the 2 sexes in the same organism. Apparently, the presence of male adds to the diversity in the DNA. But basically, they are hermaphrodites, so they can fertilize themselves. And they can cross with another worm, and once they cross, both are fertilized. The advantage of that is that once you have something genetically modified in one single worm, it's easy to get babies because there's self-fertilization. There's cross-fertilization with another worm, and there's self-fertilization. So once you have one that is modified, all the babies of that one will look the same. If you have one that is modified and you take one and you put it on a plate, 3 days later you have a lot of worms that are descendants of the first worm, and they look all the same. They have exactly the same genetic material. So that makes it very easy, again, to work in the lab with. This is the basic structure of the worm, and as you can see, it's transparent. You can see this is a digestive system. Basically, it's a tube, an external tube, an internal tube that is the digestive system. This is the mouth, the pharynx, and the intestine all the way, and the anus here. And then the other big system is the reproductive system, that is this big long thing here, where it has the gonad part and the uterus with the self-fertilized eggs inside the worm. When these eggs are laid, they are already worms growing inside the uterus. You can see here, I don't know if you get to see, there's one egg here with 4 cells. I'll show that in a later picture. But basically, it's a very simple organism. It has many of the genes that we have, and it's very simple. It has few systems. You don't have to worry about blood. There's no circulatory system. There's no heart because there's no circulatory system. The nervous system is very basic, has a few neurons. So you can study the function of neurons in a very basic system where 2 neurons connect, and how they connect and that kind of thing. So here is typical C. elegans, alive, and we can see everything through. Can you see that again? All the eggs here, the uterus, the lumen, the inside of the intestines there. Okay? Another thing here is one egg that is being fertilized and is dividing. So you can see everything in real time until the worm starts forming. Let's see if it gets long enough. Here starts the indentation, here starts moving. Gastrulation is starting to happen. And here it is, there it goes. And you have a worm that is moving like crazy before it hatches. And you start seeing all the intestines, you start seeing the intestines and everything inside. So this is a very fast video. It lasts about like 12 hours. But basically you can follow all the cells moving in real time and you don't have to kill the organism. You can track them and see what's going on through the animal. That's a huge advantage. Want to see that again? There it goes. From one cell to the whole worm. And you can see the nuclei, and you can see, now you can't see mitosis anymore because it's hard. But basically, if you're in the lab and you want to understand what cells are making this part of the worm, you can sit there day after day and with different organisms and really figure it out without needing to do much. Need a lot of patience and track one after the other one, but you will know that that cell will go there after you study it. They can trace 1,000 cells and that's it. So basically you can use this organism to study very basic things like the morphology of the digestive system, how does the nervous system develop and in what stage and what are the molecules that are associated with that. Now the second story. And the second story is about this other organism. It's called Aquaria victoria or crystal jelly. And this guy, these are the pictures. These are just contrast pictures of Aquaria victoria. This is a jellyfish that has a characteristic that is very, very interesting. It has bioluminescence in the border of the umbella. This is called the umbella. And in the border, there's bioluminescence happening in some cases. They still don't know when it happens. They can have this crystal jelly in the lab, and they have tried all kinds of things like moving the water or stressing the jellyfish or something to see when is that bioluminescence happening. And it's not clear yet what is associated with this bioluminescence appearing. What is the stimuli that gets that happening? But basically, a scientist working with this crystal jelly decided to isolate the principle that gets bioluminescence happening. And this is the part of the story that I like to tell you. Basically, in the border here, you have photoreceptors, and yeah, there are two processes happening here that produce the light that you see. You get to see green light, but there are two chemical processes happening, and you need two proteins for that to happen. The first one is bioluminescence. That is the production and emission of light by a living organism as the result of a chemical reaction during which chemical energy is converted to light energy. So it's chemical energy to light energy. This happens through this protein that is called aquarium. And this process is bioluminescence. The second process happens with a protein that is called green fluorescent protein, and its fluorescence is different than bioluminescence. In fluorescence, you have molecular absorption of a photon that triggers the emission of another photon with a longer wavelength. There's no chemical reaction here happening. Here is chemical reaction that converts chemical energy to light. In this case, it's absorbing light of a different wavelength and emitting light in a different color. Okay, I'm going to explain this a little bit more, but it's a two-step process here. In bioluminescence, the first one, what happens is that you have a substrate that in presence of oxygen gets oxidized and form an inactive product that is called oxyluciferin. This reaction is catalyzed by an enzyme called luciferase. And what happens is when this happens, there's light emitted. So in this case, aquarine would be the kind of the luciferase happening here. The substrate and the inactive product is the conformational of it changes conformation and produces light in the presence of calcium and oxygen. In the jellyfish, you have aquarine, and is generating luminescence. This light, that is blue light generated by aquarine, is absorbed by green fluorescent protein that emits in a different wavelength. So basically, the first protein produces blue light, the second protein takes that blue light and emits green light. So basically, you are seeing green light. Why nature generated this? Who knows? And basically, it's now being used for a lot of chemical engineering things. The original function, I don't think it's been understood that much in terms of why. Why is two steps to generate two types of light and not just one? So these two proteins were isolated by this guy that just won the Nobel Prize for Chemistry in 2008, Osamu Shimomura. And basically, he was very interested about this fluorescent thing, green thing that the jellyfish were having, and dedicated his life. He was working in his garage, in his basement or in his garage, and he decided to isolate that. He got that. Here it is. And by trying to isolate this green thing, he isolated aquarine and green fluorescent protein. Basically, he was after aquarine first, and green fluorescent protein was isolated as a byproduct. In the moment that he isolated this, he didn't know. It was just curiosity, out of curiosity. What is it? How do they work? And he tells a story about how he realized how this aquarine works. He was isolating this for days and days and days, and he wouldn't be able to make it light. And he wouldn't be able to make it shine or anything. And at some point, he was so frustrated that he threw everything away in the sink, and it light. It got blue light. And then he started trying to replicate that, and he couldn't do it. And basically what was going on is that in the sink, there were a lot of deposits of calcium, and this protein needs calcium to be able to work. So then he started figuring out, okay, what's in tap water that is allowing this thing to glow when I throw it in the sink. So he traced back what was in the sink, and it was calcium. So he figured out that aquarine needed calcium to turn into blue light. Now, this guy didn't know why these things could be useful, but then there were other guys that thought, well, we have two proteins. One protein produces bioluminescence. We have many proteins that do that you need a substrate, you need an enzyme to convert, and then you have light. But the second protein was more interesting because it doesn't need anything but light of one color to come in to produce light of one color coming out. You don't need a substrate, you don't need calcium, you don't need oxygen, you just need light. So when other people saw this, it was like, okay, this is a protein, it's called green fluorescent protein. Every protein is coded by DNA. So basically, if we can sequence the DNA that encodes for green fluorescent protein, and we can manufacture it, we will have a protein that is a light bulb. Whenever I shine blue light on it, it will emit green light. And it's a protein, so why not? And with this idea, that's what they did. Basically, they sequenced it. They were able to produce green fluorescent protein, and not only they were able to produce green fluorescent protein in the lab, But they were able to insert this light bulb into another protein. So make a chimeric protein. So now you have your protein of interest, example, maybe myoglobin that is in the muscles. You have your protein, and now you have green fluorescent protein attached with a light bulb. Or a protein in the cilia where now you can see where it's moving and how it's moving. But now you can see because it's producing light. The way they did that is you have a gene, the DNA that encodes for the protein of interest. Let's say it's a transcription factor in the nucleus, whatever transcription factor. This gene has two sections, a promoter, that is where all the factors attached to start reading that gene and producing RNA, and the sequence of the gene itself, of the protein itself. So now what you can do, and this is the protein that you have, this transcription factor I was talking about. So now I know the sequence of green fluorescent protein, and what I do is I cut this here, and I add the sequence for green fluorescent protein at the beginning there, and now I have the normal promoter, the sequence for green fluorescent protein, plus the sequence for the gene of interest, the transcription factor. When the cell comes and reads this and produces a protein, what I have is a chimeric protein that has my protein of interest with the perfect structure, plus a light bulb attached. Isn't that pretty interesting? So now what with that? Yeah, I have something with a light bulb. What can I do with that? And so at that point, this is a parenthesis. I'm going to tell you what we can do with that. But at that point, we had something that was green. It was a green fluorescent protein. Green. It's great for something that is transparent. It's great for something that doesn't have blood. Remember when Marco at the beginning played with the two lasers, the green and the red? So with red, I can see through. With green, I can't see anything through. So if I have something that fluoresces in green, and I put it in my finger, would I see anything through? No, because hemoglobin will absorb the light that is coming in green color, and I will not see anything. So the green fluorescent protein, it was like a great thing for certain type of organisms, but green would not work with all of them. So then this guy came on board, and he decided to modify the green fluorescent protein and generated all these different fluorescent proteins in different colors. So basically now we have a battery of things that we can make shine in different colors and use in different organisms, depending what light is being absorbed. Now we have a ton of these fluorescent proteins that have been used for different things. And now you can have even two, one protein with a light bulb that is red and one protein with a light bulb that is green, and see how those get together and do stuff. So I told you about the story of the C. elegans, and I told you about the story of the green fluorescent protein. Now let's put them together and see what we have here. This is the kind of thing we get. And these are C. elegans that we had in the lab, and in this video we took with the camera that we have in Sacramento. C. elegans alive with a light microscopy. There's a filter that will come in green pretty soon. And the visible light will start going down, and the ultraviolet light is going to start going up. So you're seeing this. Can you see these little light bulbs that start shining? Can you see this? And now the visible light is going down, down, down. They become like Christmas trees. So these guys are alive, and they're functioning, interacting, and doing everything they normally do. Now they have a protein in their nucleus. It's a transcription factor that lights up. And you can, because they are transparent, you can follow and see what they're doing. That's one example of how you can use green fluorescent protein. This is the type of worm we were using. It's the protein we are targeting is SIR5, is transcription factor. This is another example where we saw originally This is the fertilization of an egg. When you have fertilization in these organisms, remember that they are hermaphrodites, so the cells look very similar, the two cells, masculine and feminine, that come from the same organism. They look very similar. So the fertilization happens when these two cells come together, and what happens next is that the nuclei need to fuse. Once they fuse, they combine the DNA, and then they start dividing by mitosis after that. So then after that, all the cells that you have exactly the same DNA. Without that initial fusion, you don't have fertilization, you don't have the correct amount of DNA. So what you're going to see now is fertilization and these two pronuclei fusing together. They come together, they fuse, they combine the DNA, and once it's combined, we start doing mitosis. And here we have the mitotic spindle pushing in the different directions, pulling in different directions, have the first two daughter cells that are identical. And now it continues, and we will have 4 cells. No, not long enough to have 4. But here you have the first mitotic division. Now, what do we do with a green fluorescent protein? How do we see that? And this is, the mitotic spindle is made of tubulin, the same protein that made the tubes of the cilia. So now we have tubulin that is labeled with the light bulb, with the green fluorescent protein. And we have the same thing. Mitotic spindle being formed, pulling from the center, pulling the genetic material out. And you can see it in real time, what's going on, how it happens. Another example, laminin. Laminin is a protein that is in the base of the nuclear envelope, the nuclear membrane, and it has to fall apart and reassemble in this process. Because these pronuclei are coming together, it has to fall apart to allow the material to fuse. And then when the two nuclei separate, they have to reform to form again the correct shape. There we go. Come together, two pronuclei coming together, it kind of, you see, you lose the laminin, and then it starts coming back when you have the two daughter nuclei. And you can follow exactly how that happens. Every time they divide, it disappears and then it reappears, forming two.
Why does it disappear?
Because it gets degraded and it gets produced again. So to this, it gets degraded. The whole nuclear envelope destroys, gets like, and then the cell has to produce new to reform the nuclei. Histones. Histones are proteins that are attached to the DNA to form the chromosomes, the chromatin, and to organize the DNA in eukaryotic cells. Part of the high organization of eukaryotic is that they have histones in comparison to prokaryotes that don't have anything. The DNA in prokaryotes is simple, short, not too much trouble. In eukaryotes, you have much more DNA that needs to be organized to be able to work together. So histones are doing that kind of organization. And here you have labeled histones in the nuclei. All the nuclei looks shiny because you have DNA. The chromosomes are formed. Do that again. Let's see if it continues. There it starts compacting, forming the chromosomes, and the chromosomes separate in the two new nuclei. And again, every time. Want to see that again? You have all the DNA that is spread. It gets compacted to form the chromosomes to be able to separate the two different chromosomes in an organized way. If you don't compact the DNA, you can't separate it into identical parts. Okay, the chromosomes, first thing is you duplicate that DNA, Then you compact that DNA to form chromosomes, and then you separate those identical chromosomes. One goes to one daughter, and the other one goes to the other daughter cell.
What's that stray DNA? It's just kind of like,
What is what?
there's like a little piece of like stray DNA. It's just kind of,
Those are the chromosomes aligned in the plate of the mitotic spindle. Once you have So the first part you have is all the DNA, all tangled, it's spread, it's not compacted. The next thing you have is you start seeing these accumulations of DNA and still very unorganized, but you start seeing points that are a bit darker. That's what's happening. The DNA is starting to pull and to organize and tangle in chromosomes. Here they are, all the chromosomes are extended. Here they start to pull and to organize. Then the next thing you have larger points. And then the next thing you have is these chromosomes are pushed into the center. And basically you have a plate, a mitotic plate in the center. In this side you have one chromosome that will go in this direction, and this side is the identical chromosome that will pull in the other direction. First you need to organize. So that straight thing that you see is the mitotic plate. All the chromosomes.
straight. As like just like an extra little like. Because when you.
Let's see.
There are 5 below.
Where?
Upper left.
Here? Here?
Yeah.
It's probably another cell there.
But then it like comes down
No,
and
it's-
goes the other one.
I don't know.
See, it just kind
Yeah.
of comes from nowhere.
I don't know what that is, to be honest. But did you see what I was saying? The plate and then the separation. And here's another example of the same kind of worm about to be born. And we call this fluorescent protein, shining. It's very interesting when these worms are about to hatch, they start moving like crazy, like, and they move for many hours before they hatch. And you can see exactly where it goes. Basically, you can trace These are the 3 guys that got the Nobel Prize for this work. Osamu Shimomura, that was the one that isolated the green fluorescent protein for the first time, but he had no clue that it was going to be important. And this is one of the great examples about how basic biology can be taken to applicable situations. It was very basic. He was interested. He just wanted to know what was going on and what was making that shine. The second guy is Martin Schaffel, is the one that first introduced the green fluorescent protein into C. elegans and was able to track a phenomenon. And then Roger Tien is the one that has modified all this green fluorescent protein and produced all kinds of colors of fluorescent proteins that are now being used in different applications. So what? That's about a worm. We still have a lot of basic biology going on, how the nucleus separates, breaks apart, mitosis happens. So how can this be applied to larger organisms or more complicated organisms? And I'm going to give you just an example of that. Next class, we're going to see more of that. These are different examples of how fluorescent proteins are being used. This is the C. elegans, again, as I said, to track cell functions or cell proteins, whatever is happening. Glowing fish, you probably have heard about this before. Now they are forbidden in California, but people sell these glowing fish. Basically, they have inserted glowing fluorescent proteins of different colors in these fish, and you have some blue light in the aquarium, and they glow in different colors. And this has generated a lot of ethical discussion about what are we doing with genetically engineered organisms, and what are we how are we using them? Is it making sense really to do this for fun? Another example here is a tumor in the rat that the tumor cells are being transfected with a red fluorescent protein, and the endothelial cells, that are the cells that are forming the blood vessels, are transfected with green fluorescent protein. So now you can observe exactly how these intermingle and how the blood vessels are growing into the tumor to allow that tumor to grow. As you probably know, a tumor can only be very tiny unless there are blood vessels that come in. And the reason for that is that you need oxygen and nutrients to come into the tumor, and things to go out, like CO2 and things that don't work metabolized to go out of the tumor, to allow the cells to duplicate and keep growing. That has a limitation, and it's the diffusion rate of all these things that limit the growth of the tumor, unless there are blood vessels coming in. Once blood vessels are coming in and are allowing everything to come in and out, the tumor can explode and really grow in a non-controlled way. That's one thing. And the other thing is now cells can detach from that tumor and metastasize by entering the bloodstream and going somewhere else and starting a new tumor. So the idea of how endothelial cells interact with cancer cells is very important. And this is how we can, one of the things of how we can do that. For example, you can have rats with certain tumors, and you are testing an anti-angiogenic factor. Angiogenesis is the growth of, what I said, the growth of blood vessels into the tumor. Now you have an anti-angiogenic factor that tries to inhibit the blood vessels to come in. So you have rats with tumors, and you start treating them with these different doses of the anti-angiogenic factor. Basically, what you can do is, like every 2 or 3 days, you image the rat, and you can see what's going on because you just have light. You don't have to kill the rat. You don't have to take it to the pathology. You don't have to fix and take it to the microscope. The same rat can be followed over a period of time to see what's going on with that dose. You can see if you inhibit the blood vessels and the tumor goes smaller or not, or if it's not working. Another example here is using different fluorescent proteins, different colors, here green and blue, to label different neurons to see how they connect and where they are doing synapses and what's going on. Basically, now there are pictures of the nervous system using up to 5 or 6 different colors to see how the neurons are forming those connections. And I'm going to talk about this one next class. I think it's time to close. Now we have mammals that are being transfected with green fluorescent protein, and it's getting interesting. So next class we talk about that with more examples. Do you have any question, comment about this? Have you seen about this green fluorescent protein thing before? You probably heard about the green pigs a while ago. So that's how it's been done, is by understanding and being able to decodify to sequence the code for proteins that have special functions, and now being able to insert that function wherever we want. In this case, we're talking about something that glows. You can insert proteins with other functions like enzymes. You can tag an enzyme to a protein that you want this enzyme to end in certain compartment. So now I can tag it to a protein that I know is going to go to that compartment, and then I make it go and see what happens. All kinds of things can be done. So next class we talk about genetic engineering things and how it's been done and what are the trends going on. Okay. See you next class.